Hello, everybody. This is Miss Peachy from WCA Biology A, and I'm going to do an overview of Unit 2, Lesson 4. So, I encourage you to do the reading before you actually watch these, just so that you are kind of having a bit of a background before you come to kind of my explanation of the unit. This one is a bit of a tricky part. There's a lot of information here, and it goes through it fairly quickly. So bear with me because I'm going to do it a little bit differently to try to illustrate the same concepts to you, and hopefully in a way that is a little easier to understand. So essentially what this lesson is covering is a process called protein synthesis. Protein synthesis is very important because it's the process that your body uses to read the information that's found encoded in your DNA and then to make proteins from that. And proteins are used in so many different aspects of your body, from different proteins that make structures, like the lesson refers to the protein keratin. Keratin is the protein that makes your hair and your nails, and it's found in your skin and stuff like that. To also, kind of a functional part of your body as well. So certain proteins are used to help. It also refers to a protein that helps to clot your blood so that if you get an injury that you don't bleed to death, right? So there's so many different proteins and they do so many different jobs that they're very, very crucial. So the process of protein synthesis is super important as well. In a eukaryotic organism like we are, protein synthesis begins in the nucleus of your cell and it ends in the cytoplasm on a ribosome. So let's refamiliarize ourselves a little bit with the structure of the cell here. And remember I said that there are, in the cell, that we have the nucleus. We also have other organelles, which we really didn't talk a ton about. But in that nucleus is your DNA. And I'm just going to write just a little. I'm terrible at drawing the DNA. It's found in there. As you exit the cell, there's a structure. Whoops, it's a little bit fat. Called the rough endoplasmic reticulum. And it's kind of found right next to the nucleus. On the rough endoplasmic reticulum are these little things called ribosomes, and they kind of are all over. So it's called rough endoplasmic reticulum because the ribosomes are bumpy, kind of like sandpaper, and so it's rough in appearance. It looks rough. Okay, so here we have, if I can try to label these again, those little black dots. Oops, make this a little skinnier of a line. Actually, let's do a different color. These little black dots here, those are ribosomes. Okay. So the process of protein synthesis, which is what we're learning about, and I'll title this up here. I love how my writing looks like I'm a kindergartner. Has two main steps to it. The first one is transcription. Transcription is, it takes place inside of the nucleus, okay? So let's see if I can write this really small inside the nucleus. I doubt it because I'm really bad at trying to write tiny on this thing. I'm going to just zoom in and see if I can. The problem is when you zoom in, sometimes your writing gets really fat too. Okay. Not too bad. And then we have the second process, which is translation. And translation is going to take place on the ribosome. Okay. So we have transcription and translation. Transcription is where DNA is read and messenger RNA is made. Okay, so inside of the nucleus during transcription, DNA doesn't turn into, but it is read. Again, I'm going to zoom in just a little bit here because I just don't get very good penmanship if I don't. So mRNA stands for messenger RNA. Okay, that's what happens with transcription. Translation, the messenger RNA is read. Whoops. And amino acids are made. Or they're not made, but they're amino acids are put together into one long chain. Okay. So let's put this on the screen for you here. So basically, when we have a protein is a chain of amino acids that are put together in order, and they go in a specific order. And when you look down here, here's an example of part of the protein for keratin. Each one of these is amino acid, and each one is put together, and it has to be put together in the proper order for the protein to work correctly. Once the chain is assembled, then the protein is folded or bent into a specific shape, and that kind of completes the process of protein synthesis. We're basically talking just about the transcription and translation today, which is to assemble this protein or this amino acid chain, which is called a polypeptide chain. We're not really going to look at how they fold them or anything like that. Okay, so let's look at transcription. So this is happening inside of the nucleus right now. Now we're going to go in the nucleus. So here we are inside of our nucleus. And what happens here is the DNA is unzipped into two separate pieces. Remember, DNA is a double helix, so it's unzipped first, and it's got two sides to it. Kind of exposes all the different nitrogenous bases in here, right? So step one is the transcription. Step one, DNA is unzipped. It looks like a zipper, that's why they call it that. Okay, so only one strand of the DNA is read. So let's look at the strands. So remember, we have T, A, C, and G. That's my G, it's not a very good G. And the complementary strands, so remember the opposite side of DNA doesn't exactly match, it complements. So T always is going to match up with A. A is always going to match up with T. C is always going to match up with G. G is always matching up with C. A with T, T with A. Okay. So this is kind of what it would look like when it gets unzipped. So only one half of it, or one strand, is read. It's read by an enzyme known as RNA polymerase, which There's a lot of information here, and I just feel like that one's not the most crucial piece, but I'm going to put it in parentheses here just so you know. So that's the job of an enzyme called RNA polymerase. Okay. It's supposed to be an R. It looks like a V. But I need a better stylus. All right. So what RNA polymerase does is it makes a strand of messenger RNA. Messenger RNA complements the DNA, but it has a slightly different base pairing, right? So normally, let's say RNA polymerase is going to use the pink strand up here as its template. If it were making DNA, it would literally just make this. But it's not making DNA, it's making RNA. Since it's making RNA, there's some slight differences. So let's outline the difference over here between RNA and DNA. So here we have RNA, and here we have DNA. These are our two types of nucleic acids. RNA is a single-stranded. Okay, DNA is a double helix, it's double-stranded. So there's our one difference, right? Second difference, RNA uses a sugar called ribose sugar. DNA uses a sugar called deoxyribose sugar. Okay. Third difference is the bases. RNA uses, or also DNA, we know DNA already, adenine, which normally is going to bond with thymine, right? And cytosine, which normally bonds with guanine. RNA also has adenine. I'm just going to put an A because I'm going to run out of space. But adenine doesn't bond with thymine. Instead, it bonds with something called uracil. It still has cytosine and guanine. Those are the same. So the only real difference is in the base pairs. I'm just going to put a green here, is uracil. Uracil is different. Uracil instead of thymine, okay? So going back over here, when this RNA polymerase is making the messenger RNA, it doesn't make ATGCTA. It doesn't make that because that's DNA. It will basically assemble the same thing that the yellow side is, because remember the pink is the template. The yellow side is kind of the matching or the complement. Instead, it's going to make, wherever you see a T, you're going to substitute a U. So its messenger RNA will be a UGCUA. It's basically the same thing. It's just that, it's literally everything except the T's are substituted by U's. So this is our single strand messenger RNA. It doesn't have a double strand. So this thing up here then, by the way, this DNA will just, it'll come back together. It'll just wind back up and it'll be done. And then this little guy, oops, it was in the nucleus. Now it's going to wiggle its way out of the nucleus and it's going to find one of those black ribosomes. So that was transcription is completed. That's all it is. Transcription is unzipping it, using one of the strands as a template to make the messenger RNA. That's the complement to it. Once that messenger RNA is made, it leaves the nucleus and goes to find a ribosome. And now we're into the translation part. So with translation, actually, you know what? I'm just going to put a step 4 here just so that we have that mRNA leaves nucleus to find ribosome. Okay, now we just so we officially write that part down. All right, so now we're out here in the cytoplasm. Well, most of the ribosomes are attached to the endoplasmic reticulum, but some of them do just freely float in the cytoplasm as well. So it doesn't really matter where; it just has to find a ribosome. Ribosomes are kind of cool. They're like little structures, kind of like, I don't know, they got like a top piece and a bottom piece like this. I'm going to draw the bottom piece a little bit skinnier. So those little circles are a little bit more complicated, almost like a little sandwich cookie or something. So this is our ribosome. Pretend like it's just shaded in there. So now this messenger RNA, which we made before, it's AUGCUA. Okay. Just going to write it out here. Okay. It's going to get fed through that ribosome, kind of like, I don't know, kind of like a piece of fabric getting fed through a sewing machine, or, you're laminating something, or I don't know, whatever. Use whatever example you want there, but it's going to get fed through, and it's going to be read 3 bases at a time. Every 3 bases is called a codon. Called a codon. That's what that is. So AUG is the first codon. Then another molecule known as tRNA, looks like a little T, like that. They're like little alien ships that just kind of fly around. Each one of them is carrying an amino acid on the top. It kind of looks like a person. And the bottom of it has a sequence of code that's the complement to codons. So the complement of AUG would be UAC. Remember, with RNA, A bonds with U, C bonds with G. So the complement here is, so I can make sure I have the small one. So it carries like the complementary code, UAC. It recognizes that it is matching this code. So it knows if it matches, or if it has the complementary code, it's going to leave behind this little blue ball, which is called an amino acid. By the way, this code UAC that is complementary to our codon is known as the anticodon. So UAC is the anticodon, AUG is the codon. They match each other. They're complements. Remember, they're not exactly matching. They're just the complement. So UAC has a very specific amino acid, cysteine, which we're going to abbreviate CYS up here. And it leaves it behind. So now, once it recognizes, ooh, I match you, now it's going to leave this little blue ball behind. And then the rest of it is going to fly away and find some more. So it's like, all right, I'm just going to fly away. And this part stays attached to it, by the way. I'll go find another cysteine for a different, because they're constantly busy kind of doing their things. I don't think I can, somehow I can't delete the red. So this is left behind. This strand continues to get fed through the ribosome to read the next. So our next codon, CUA. And then another tRNA molecule comes into play. And this tRNA molecule is going to have the anticodon to match CUA. So it will have G, A, U. That's how it knows that it matches the next one. And it will leave behind another amino acid. Let's see what it is, because I don't know all these off the top of my head. Leucine. And we'll color that one, I don't know. What color should we make it? Like a little red ball. And it will leave that one behind. And this just keeps happening over and over again. The little messenger RNA strand will forward. It goes like 3 at a time, right? So it's going to move forward another 3. Another little transfer RNA molecule, tRNA molecule, will come in. It will have the anticodon at the bottom that is complementary to that codon that's being read, and it will have an amino acid on the top. And if they match, it will leave its amino acid behind. And it does this over and over again until it forms a long chain like this, all in a row. And the very last one that's put into play, or the very last codon that's read, actually is what's called a stop codon, and it will tell, like basically tell translation to stop at that point. So then it will cut it off, and it will be the last amino acid that's read in order. Now this picture, I keep kind of going back to it in your lesson. If you go all the way down, it goes through this chart. This chart's going to show you all the codons and which amino acids they code for. So remember, codon is 3 bases in a row, 3 RNA bases. And every strand of messenger RNA is read 3 bases at a time. Each 3 bases is a codon. When you read this, you read it like this. You start with the left side. That's the first base in the triplet. The top is the second base, and the side is the third base. Let me just make this bigger. And I'll zoom out a little bit so you can kind of see this. Okay. So basically, if I have the amino acids, say UGC, I start on this side and I find U. And that tells me that I have to limit my search now to the top row only. My next letter is G, so I'm going to go to the top and find where the G is. So now since I'm in the top row, I'm only limited to this square because this is where G is in that row. My last one was C. So I go down the side and I find C, and whatever is here, UGC, CYS, that is the name of the amino acid. It's actually the abbreviation for the amino acid, which is cysteine. So if I have the base sequence UGC, then my amino acid is cysteine. And as you can see, if I have the base sequence UGA, it says stop instead. So again, if that would occur at the end of a polypeptide chain, which is a chain of amino acids, that would tell translation to stop and just cut off your chain right there. It's called a stop codon. Okay, so yeah, it's pretty cool. This isn't my favorite. This will tell you the abbreviations and what they are down here. There's another kind of chart that you can use that's a circular one, which I'll show you in our lesson if you come to my live lesson. Okay, so that's basically it. To review, protein synthesis is transcription followed by translation. Transcription takes place in the nucleus, where DNA is unzippered. It is read, one strand is read by RNA polymerase. It uses that strand as a template to make a strand of messenger RNA. Remember that every time you think you should put a T in, instead you should put a U in when you're making messenger RNA. That messenger RNA then leaves the nucleus, goes out to the cytoplasm to find a ribosome. When it finds a ribosome, it is fed through the ribosome and it is read 3 bases at a time. Every 3 bases is called a codon, and each 3 bases is going to code for a specific amino acid. A molecule known as tRNA, or transfer RNA, contains an amino acid on one end and an anticodon on the other end. The anticodon is complementary to the codon being read, and so if it complements, then it knows to leave behind its amino acid, and a bunch of these tRNAs just keep coming in and they form like a whole chain of amino acids, which we call a polypeptide chain. Once you get to the end of it, then a stop codon is read. Protein synthesis stops, and then we get to the point where this whole chain gets folded up into a protein eventually. Okay, so that is pretty much protein synthesis in a nutshell. Hopefully that made sense, ladies and gentlemen, and let me know if you have any questions. Otherwise, I will see you in the next lesson.